Okay, so the big three methods of cellular transport are diffusion, osmosis, and active transport. Diffusion is the most important. Okay? The reason diffusion is the most important is it's the one cells use the most. Okay? It is the only way that stuff moves in the cytoplasm of a cell. Okay? There's no like trucks or something like that that move things around from one place to another within the cell. Okay? There's just diffusion. The really good thing about diffusion is that it's free. The cell requires no energy for it to happen. Right? So it's like, when you order something on Amazon, as long as you're okay to wait 2 days, you can get free shipping. Okay? Or you spend over $35 or whatever. Okay? As long as you're willing to wait, okay, you can get free shipping. Well, the cell is very much that way. Okay? The cell is all about free shipping. If the cell doesn't have to spend energy, then it's not going to. Now, diffusion has some drawbacks. Well, let's talk about the positives first. First positive is it's free. Okay? It's free shipping. Second thing is that it will naturally move materials from an area of high concentration to an area of low concentration. Okay? And this is just a natural process. The cell actually can't control it. It just happens. Okay? And it happens everywhere, not just within cells. Cells just happen to harness it. Okay? But the same thing happens with a fart. Okay? If somebody farts and you're nearby, the experience for you is far more intense than it is for someone who's on the other side of the room. Okay? Because the gases within the fart are going to diffuse throughout the air in the room. Okay? But diffusion doesn't work very well over large distances because it spreads out very quickly. Okay? So like I said, if you're nearby, you're in this area where it's very highly concentrated. Okay? But very quickly, it becomes less and less concentrated the further away it moves. Okay? So, diffusion's not very fast. Okay? It's also not going to work over long distances. Is that okay for a cell? Yeah. How big is a cell?
Not big.
Not very big. Okay? So diffusion works great because cells are generally pretty small. Okay? It's also why cells do need to divide as they grow. If they get too big, this process won't work efficiently enough for them to transport materials. There's kind of an upper size limit. We're going to talk about that next week. Okay? An upper size limit to cells because of diffusion. Diffusion is the only way they can transport materials. They can't control it. Okay? It's kind of slow, and it only works over short distances. But that's okay because we're talking about a really small volume in which diffusion has to work when we're talking about an individual cell. Okay? If we were relying on diffusion to transport materials from even the distance from our heart to our brain, how well would that work? Not very well. Okay? We have to have an active system, our circulatory system, that pumps blood, and then, yes, diffusion still happens, but blood is quickly pumped to the brain, and then diffusion can work over the small distances between cells in the brain. Okay? Because our blood vessels kind of go everywhere. Right? So we don't make diffusion work over the length of our body or even half the length of our body. Okay? We only make diffusion work over cellular distances because we help it along. Right? That's why single-celled organisms generally don't get very big. Okay, so simple rule of diffusion, okay, is this. Okay? In the absence of other forces, a substance will diffuse from where it is more concentrated to where it is less concentrated. We call that down the concentration gradient. Okay? Kind of like stuff flows downhill. It goes down the gradient. Okay? To make something go from an area where it's less concentrated to more concentrated actually requires energy. It's like pumping water uphill. Okay? So diffusion is just the natural tendency of things to want to spread out. Okay? And that will naturally take them from where they are highly concentrated. If we're talking about within a cell, that could be wherever they were produced, to the areas where they're needed. Because the areas where they're needed, their concentration is going to be low. So they're naturally going to move there by diffusion. Right? That sort of makes sense? So diffusion works really, really good because it's free, it works over small distances, it takes things to where they're supposed to go. Now, drawback of diffusion. It's not targeted. Okay? It just kind of naturally goes to an area where it's less concentrated, which is probably where it's needed, but it might also go other places. Okay? It's going to areas of lower concentration. That means all areas of lower concentration. So rather than being like a single bullet being shot, it's more like a shotgun approach. Okay? A lot of little pellets flying everywhere. Some of the pellets are going to hit the target and some aren't. Okay? But still enough of it is going to get to where it's needed. So it works. Okay? Everybody with me on that? So diffusion, good, free, slow, not targeted, okay, but effective. All right. Diffusion is also known as passive transport because it requires nothing from the cell. Okay? The cell expends no energy. This process just happens. Okay? And that's, of course, why all cells on Earth have been using this process for billions of years. Why would you go away from something that works, especially if that thing that works doesn't cost you anything? All right. Questions on diffusion? Diffusion, most important process. Now, the thing with diffusion is, this is just for transporting inside. Okay? The stuff is already in the cell. It's not how it gets in. It's already in there. It's going from one place to another within the cell by diffusion. Okay? So it's intracellular. Okay, second most important process is the process of osmosis. Okay? And osmosis happens on the membrane, or within the membrane, or across the membrane, probably is the best way to say it. It happens across the membrane. Okay? It is the way water, and water only, okay? That's the key thing. It's the passive transport of water. Okay? Only water moves by osmosis. How many people have heard the term osmosis before? Okay. And you probably heard it.
Grade 7.
Yeah, grade 7. Okay. And in the context of how to get water very clean, right? You probably heard it in the context of reverse osmosis. Okay. Lots of bottled water, okay, Culligan water. That's all reverse osmosis, filtered water. Okay. Now, reverse osmosis is an active process. It's artificial. Okay. Real osmosis is a passive process. It just happens. A cell cannot control it. But it usually results in the movement of water, okay? Or always results in the movement of water, okay? So if I have a situation like this, so I have a U-shaped tube. This is actually something you'll do in Bio 20, okay? And I have a piece of membrane across the middle of it. What we usually use for this is sausage casing, okay? Which is made of intestines, yeah, which is made of cells, okay? So it's essentially just a big piece of cell membrane. Okay? So we put it across the middle. On one side of that membrane, we have a highly concentrated salt solution. On the other side, we have a very low concentrated salt solution. What we see happening because of osmosis is not the salt moving, okay? Because the salt, if it could, would move by diffusion until the two were equal. But it can't. The salt can't get across the membrane, but water can. So water actually goes the way opposite the salt would go. Okay? So it moves across the membrane into the area where the salt is highly concentrated and the water is less so. Okay? So it will move until the water level rises high enough on the other side that the concentration of salt on both sides is equal. Okay? So it's a passive process. The cell can't control it, but it happens in all cells. Water moves across membranes until salt concentrations are balanced. Okay? That's good and bad. It's bad if you don't have access to fresh water. Okay? You guys heard the expression, water, water everywhere, not a drop to drink. It's what people say when they're stranded in the middle of the ocean. You know what kills people when they're stranded in the middle of the ocean? Dehydration. Yeah. Okay. Dehydration is what kills you if you're stranded. I mean, if you have a boat, otherwise it might be something eating you. But okay, if you're stranded in the middle of the ocean, you have a boat, you're gonna likely die of dehydration. Right? Because what will happen is you'll run out of fresh water. Okay. Your first option, once you run out of fresh water for something to drink, is what? Your urine. For the first day. Maybe 2 days, your urine will have less salt in it than the ocean water. Okay?
Yeah, sure.
There's actually some really weird, like freaky diets out there where people drink their own urine.
It's,
That's
yeah.
best.
Yeah. But don't do that. It's just a hygienic thing. Okay. But if you were if you were stranded in the ocean, those first couple of days, it would be wise to save your urine. It would actually be okay to drink. The issue is, as you become more and more dehydrated because of sweat, okay, losing water that way, your urine will become more and more concentrated until, well, it would be almost impossible for you to like physically drink it. Okay. But also because it would have more salt or the same amount of salt as the ocean water. So then, because you're so thirsty, you would probably drink the ocean water. Now, there's two problems with that. The first is the ocean is the world's toilet. Okay? Remember Finding Nemo. All pipes lead to the ocean. Okay? Gross. Second thing, it's full of salt. Okay? And not table salt, like all kinds of salts. So when you drink it, that salt gets absorbed into your blood. And the concentration of salt in your blood goes up. Water in your cells moves out of the cells and into your blood to try and balance the concentration. So drinking salt water actually sucks the water out of your cells. Okay? And then you end up actually urinating more because your body's trying to flush the salt that you've consumed out. Because osmotic pressure is resulting in more your blood essentially becoming too full with fluid, so your kidneys filter the fluid out to get your blood balanced again, and you have to release the water. Okay, so you end up becoming dehydrated. Like for every liter of ocean water you would drink, you would lose 1.5 liters of your bodily fluid. Okay, it's that bad. It's really concentrated, and the process of osmosis is what's going to pull the water out of your cells. Okay, is everyone kind of following me there? It's not something your cells can control. It just happens. It's a passive process. Most of the time, that passive process works for you. Okay? If you are dehydrated and you drink fresh water, the water is rapidly absorbed into your cells because your cells have a higher concentration of salt than your blood once you've ingested some water. Okay? So then the water will move back into your cells and rehydrate. Okay? Sometimes we can even accelerate that process by adding some electrolytes to the solution that you're drinking. So if you've ever gotten sick and you've had to get an IV, okay, just a saline IV, right? There's a small, very small amount of salt in that IV, and that actually helps your cells to get going on that osmotic process and getting you rehydrated. Okay? So if you've ever gotten an IV, that's all it is. It's like 0.1% salt solution. Okay? It's basically nothing, and your cells absorb it really rapidly because it goes directly into your blood. Okay? You usually end up with an IV if you've been like throwing up for days and you can't even keep water down. Okay? They'll put an IV in you to just get you rehydrated. Okay? That works by the same process, by osmosis, getting fluid in so your cells can fill back up. Okay? If you are looking at like plants that grow near a slough, when the slough starts to dry up, what do you see on the ground? You see the white salts left on the ground. What happens to the plants around the edge? They die, okay? Because what happens is the soil gets highly concentrated with salt, and it actually begins to draw the water out of the plants. Okay? So instead of being able to transport water from the roots to the leaves, it actually gets pulled the other way, okay, by osmotic pressure. What actually pulls the water up the plant? Okay, we talked in the chemistry unit about cohesion and adhesion being important. Okay, they're the things that essentially keep the water from falling back down. The pulling force is actually osmosis. When water evaporates from the leaves of the plant, what gets left behind? Salt. Okay, so there's salt there in the leaf, and water moves naturally towards the salt by osmosis in order to balance the concentration. Because all those water molecules are linked by hydrogen bonds, they all move up. Okay, so only the ones in the leaf actually move towards it, okay, but they pull on all the other ones. So it's actually osmosis that helps a plant move water, which means how much energy does a plant expend to move water? Zero. It's free. It's using a simple cellular transport process to do it.
What would happen? So you said saline, like if you get an IV, it's like 0.1%.
Yes.
What would happen to your body if it was like 1% instead?
If it was 1%, it might go the other way. I don't know the balancing concentration, but it's always very low. Now, they would have different solutions for different purposes. Yeah. Okay, so the issue here, guys, what we were talking about, if you drank seawater, okay, would be something like the diagram, the lower right part of this diagram. So if I have a cell, okay, in what we would call isotonic conditions. Isotonic means the concentration of salts in the cell and outside of the cell are the same. So osmosis just goes back and forth equally. The same amount of water goes in and out. Okay? If I put a cell in what we call hypotonic conditions, that means the concentration of salts in the cell is higher than the concentration outside the cell. It would be like if I put a normal cell in distilled water. Okay? If I did that, the cell would die. Why?
It's having way too much water.
Yeah. Could the concentration of salts inside the cell and outside the cell ever be balanced if I put a cell in distilled water? No. There's no salt in distilled water. So osmosis will just continue to pour water into the cell, trying to get the concentrations equal until the cell pops. Okay? Which it would literally do. It would rupture the cell. Okay? The opposite happens if I put a cell in salt water. That would be hypertonic. The solution outside has more salt. Okay? Water gets rapidly sucked out of the cell, okay, because it's trying to move towards the area of high concentration. Ever seen what happens to a slug when you put salt on them? That's it, right there. Okay? If you put salt on a slug, all the water in the slug rushes out of the slug because they don't have a hardened epidermis like we do. Okay? They're kind of slimy, so it all just pours out of them, okay, and they shrivel up.
Could you put distilled water on one end of the tube and then a really high concentration of salt on the other, and then make it so like water is going on one side of the tube?
You would get probably pretty close to that, yeah. Okay, so for osmosis, the things we need to remember about osmosis, okay, are this first sentence. It is passive. It is of water only, and it is across a membrane. Okay, it's always across the membrane. This doesn't happen from point A to point B inside the cell. Okay, osmosis happens, there has to be a membrane that's separating the two solutions. Okay, so it happens across the membrane, and essentially it's just trying to gain balance or equilibrium. Okay, it's trying to balance the concentrations of salts on either side of the membrane. Okay, so the key points: passive transport, water only, across the membrane, gaining equilibrium. All right, questions on osmosis? All right, the last of the big three is active transport. Okay, it is different from the other two because instead of being passive, it's active. This process actually uses energy. The cell expends energy in order to pump stuff across the membrane. Okay, so because it's pumping it in a direction opposite the natural direction. The natural direction of movement is always from high to low. Active transport is pumping from low to high. Okay, so active transport works differently because it is going the opposite direction. To make water go downhill requires nothing. To make water go uphill requires energy. Okay, it's the same thing here. If this is my cell membrane, so that's what's in the diagram here, this kind of middle layer, okay, embedded in the cell membrane, we talked about these things, okay, there's stuff embedded in the cell membrane, in particular, carrier proteins. Okay, you might want to circle that or make a note of that term. Okay, there are carrier proteins embedded in the membrane. They're like gates. Okay, they're like gates that can open and close and allow stuff in or out of the cell. The stuff is very small. Okay, we're not moving giant protein molecules through these gates. Okay, they're very, very small. Only electrolytes, salts, can move through them. Okay, so what will happen is, okay, the gates are normally in this position. The carrier proteins have this shape, resulting in the gate being closed. If the cell needs to very quickly move ions from outside the cell to inside the cell, the cells in your body that do this most often, muscle cells. Okay, they have in their membrane what is called the sodium-potassium pump. Okay, and in order for your muscle to effectively contract and relax, okay, every time your muscle contracts, sodium and potassium switch sides on the membrane. For it to contract again, they have to be pumped back the other way. Okay, that process happens very quickly, but it expends a lot of energy. Right? But that's how the cells, the muscle cells, can relax and contract, relax and contract. It's got the actin-myosin group there requires this balancing and shifting of ions. Okay? So they will expend energy to change the shape. When energy is applied, chemical energy, not electrical. Okay, I don't want you to think like there's this zapping process that happens. Okay, but when there's energy applied to these carrier proteins, their shape will change. They'll go from being kind of bulbous on the outside, okay, to being bulbous on the inside, which means they open up and material comes in, and then they shift their shape like this using energy, so that they're closed on the outside, open to the inside, and the material will move in. If they're doing that, they create kind of a suction that pulls material into the cell. Right? So we've got, for active transport, okay, it's the pumping of solutes against the gradient. So from low to high. Okay? It uses energy, okay, so it expends energy, okay, and I don't have anything in there about the carrier protein, but it involves the changing of shape of carrier proteins. Okay? Changing shape of carrier proteins. Okay, everybody all right with active transport? Okay, so those are the big three: diffusion, osmosis, and active transport. Okay, they are called the big three because they involve the movement of materials without massive changes of shape of the cell. Okay, there are other methods of transport which are less important, but we're going to talk about, okay, that have to do with the membrane physically enveloping stuff. Okay, that's totally different than these methods of cell transport. So generally on a unit exam, I would give you a situational question that would be asking about the big three. Okay, like one for each one basically. All right, so I might ask you something like, you know, if I, let's say, put a cell in distilled water, explain what would happen. Okay, and you would say, okay, so if I put a cell in distilled water, what's different about both sides? Okay, well, there's salt differences. So that probably means water is going to move. Water's osmosis. You follow the process? Okay, so there's that stuff I have to know, the stuff I have to memorize, and then I have to apply it. I have to, okay, how does osmosis work? Here's osmosis, here's how it works, this is what's going to happen. Okay, so it's going to be that kind of situational question. Okay? Okay, if a cell wants to move stuff out of the cell, okay, it's going to use a process called exocytosis. Okay, and if you recall, this was one of the functions of the cell membrane, to control exocytosis. Okay? The Golgi apparatus helps with this, because it's usually the thing that's making the vesicles that go towards the cell membrane to be expelled. Okay? So, a vesicle, which is basically just a piece of cell membrane that's sealed, so there's something stuck inside of it, okay, is going to move towards the cell membrane. Once it hits the cell membrane, it merges with it. It essentially becomes part of the cell membrane. And when it does that, the seal is broken. Right? So you can see here, in both of these processes, we have a vesicle that is moving towards the membrane, and when it touches the membrane, it just becomes part of the membrane, and its contents are expelled out of the cell. Okay? Two things are done that way. The removal of waste, probably the most important way for most cells, and any cells that are secretory. That means any glands. They effectively make hormones. Okay? Any cells that are part of a gland are going to use this to secrete their hormones into the bloodstream, because this is the quickest access to the bloodstream, because what's a cell going to be surrounded by? Not always blood, but certainly intracellular fluid that's connected to the blood. Okay, very closely connected to the blood. So this is a very good way to quickly get things into the blood. Because if you ever look at what a gland would look like in terms of how many like vessels, blood vessels would be in it, it's amazing. Okay, like your glands are just, there's veins in them everywhere. There's tons of vessels and veins and capillaries and stuff in them, okay, that allow them quick access for their materials to the bloodstream. Okay, so two things are going to be expelled that way using exocytosis. So the big thing about exocytosis is this is how we secrete large molecules. Okay? All the other stuff we were talking about just moves small stuff. Salts, okay, sugars, that's basically about it. Okay? Salts and sugars, okay? Which need to be transported all the time, but large molecules, they don't work so well with those other three processes. They got to be moved in and out of the cell by massive changes in the membrane. All right? So it would be kind of like this. I'm delivering something to the school. Okay? It's one box. I'm going to come in through from the front door. Okay? It's not a very big box. I can carry it. I come in through the front door. Okay? If I'm delivering enough desks for the entire science wing, I'm coming in with a what? I'm coming in with a big truck. Okay? And I'm probably going to be taking the doors off so that I have a wider area to move the big cargo through. Okay? Exocytosis is how we get big cargo out. We change the membrane completely. Okay. All right, everybody all right with that? Okay. Now, into the cell. Same process in reverse. Okay? All of these processes, endocytosis and exocytosis, all involve the membrane changing shape. Okay? Either an envelope touches the membrane, merges with it, and expels something, or the membrane folds in on itself and creates a vesicle that then moves into the cell to wherever it's supposed to go. Okay? So endocytosis, the cell takes macromolecules. Macro means big. Okay? Micro means small. Macro means big. Okay? So big molecules and particulate matter by forming vesicles budded from the cell membrane. Okay? There's actually three kinds of endocytosis, and they just have to do with what's being taken in. The process is still the same. The membrane buds in on itself. Okay? So the other day, I think I was telling you about how this is kind of like when you pick up after your dog. Right? You put the plastic over your hand, okay? You grab the poop, and then you turn the plastic inside out. You just created a vesicle. That's exactly how the cell takes in big particles. The particle will touch the outside of the cell will envelop it. Okay? You just close in around it and take it inside. This is how the amoeba eats. Okay? Like this is an amoeba here, or it's supposed to be. Okay? And it ingests food through that process. In particular, we call that phagocytosis. Okay? So if we're enveloping a solid particle, it's called phagocytosis. P-H-A-G. Okay? So there's phagocytosis, cell eating. Pinocytosis, cell drinking. Okay? So if osmosis is not getting a job done quickly enough, the cell can actually envelop large amounts of intracellular fluid. Okay? Like it'll just go. Okay? And then there's receptor-mediated endocytosis. So top left, we see pinocytosis. The cell membrane just folds in, and there's a big, or sorry, not a big, but a small water vacuole created. Okay? If we're talking about food, phagocytosis, okay, then the cell envelops the food and it becomes a food vacuole. Okay? If it's a hormone, then this process is triggered by receptors. Okay? And we said already that the cell membrane is covered with all kinds of proteins and things like that. Well, there are receptor proteins on the surface of your cell membrane for each signal that a cell can receive. Right? So it's not like a radio. A radio has one antenna that receives many signals. Your cell has many antennas, one for each signal. Okay? I know that seems impractical, but that's how your cell works. Okay? It only has a certain number of signals to receive. So it's got one for receiving like adrenaline, one for receiving insulin, one for receiving estrogen or testosterone, whatever. Okay? And when that signal is received by the receptor, the cell immediately is signaled, hey, that's important. Take it in. Okay? And it does right away. Okay, so phagocytosis looks like this. This is actually one of your immune cells, one of your T cells, okay, like the killer T cells, the ones that envelop and engulf things and destroy them. Okay, they engulf bacteria and essentially eat them, right? And that destroys the bacteria. So they create these pseudopods which envelop the bacteria. The bacteria joins with a peroxisome, okay, and that's where digestion occurs. They call it a phagolysosome, which is not something you'll ever need to know, okay? And then the bacteria is broken down and the waste materials are expelled, okay? Not actually used for food, expelled, okay? Because those bacteria probably contain a lot of toxins. Incidentally, that's the difference between a bacterial infection and a viral infection, okay? If you have a viral infection, you get sick because your body's killing its own cells. A bacterial infection, you get sick because bacteria release toxins that make you sick, which is why usually a viral infection has different symptoms than a bacterial infection.
So when the cells bring stuff in, like, where's the stuff coming from?
Usually from the intercellular fluid. So if the blood has brought something in, it's kind of like your capillaries are not a sealed vessel.
Yeah.
They're very small, but they're porous. And the pores are big enough that sometimes stuff can get out of the bloodstream into the area between the cells, and the cells won't go. Yeah. Okay, now, for these cells, they're obviously in your blood. They're floating through your blood doing that job. Okay, so that's phagocytosis, that's cell eating. So if it's a solid particle, phagocytosis.
Is this one of the ways, like, if you get carbon monoxide poisoning or something, if you have excess carbon in your bloodstream, and that's how it's absorbed into a cell?
No, carbon monoxide is actually absorbed through the membrane.
Okay.
Yeah, like not through either of these processes. That's like that's a passive diffuses, gets through the membrane, just diffuses through it, gets through. Okay, endocytosis is the drinking process. So the membrane just pulls in, same process, the cell folds in on itself, okay, but it's only taking in fluid, and in that fluid could be, electrolytes and things like that. So if a cell is really water stressed, it will probably do this when water is available. This is actually what it looks like under a light microscope. Okay, you can see, okay, that this cell, they stressed it out. Okay, they made it really dehydrated, and then they put it in a solution where it would quickly pull stuff in, and you can see there's all of these vesicles being formed all along the cell membrane as it pulls the water. Okay, and then receptor-mediated endocytosis. Okay, it's still the same process. Membrane folds in on itself, but this time it's triggered by a receptor receiving a signal. So this little receptor here receives its chemical signal, okay? The cell goes, oh, we need that stuff, take it in, it envelops it, and it goes to whatever part of the cell it's supposed to go to, okay? Under an electron microscope, it looks like this, okay? So you can actually see that in a few of these places here, you can kind of see the receptor molecules, okay? They're kind of shaped almost like what they drew here, okay? And they have on them their signal molecule, okay? Once that signal is received, okay, then it pulls it in. You can actually see it better here. You can see all the little signal molecules, okay, these kind of circles here, okay, and they're on top of the receptor molecules. The cell is just about, here's the cell membrane, it's just about completed the process of making the vesicle. Okay, those two ends of the membrane will just merge with each other. The cell will seal back up, and they'll have a vesicle inside. Okay. So those are the three kinds of endocytosis. Okay. I basically never ask about those in a written response item. They're more kind of recall in the multiple choice type item. Okay. You just need to know the difference. Okay. The difference is what causes the process. The process is the same for all of them. All right. Questions on that? All right, let's have you guys quickly answer those 4 questions. We'll go over them, and then we're going to do our pre-lab for tomorrow. Now, I remember if it was you guys in my period 4 class I was telling about Mr. Van Erf. Okay, so color blindness is caused by a person's cells being unable to produce certain proteins. So if you're colorblind, the problem is in your DNA, because color blindness is genetic. Okay? And as a result of it being genetic, it's actually 4 times more common in men than it is in women, because it's on the X chromosome. If you're a woman, you have 2 X chromosomes. It's recessive, so you're more likely to have a copy of the dominant gene and not the recessive gene that is color blindness. If you're a guy, you have an X chromosome and a Y chromosome. If you have the color blindness gene, you're colorblind because you only have 1 copy of it. Okay, so it's way more common for men to be colorblind than women, and there's different types of color blindness. Now, Mr. Van Erf, he is completely colorblind. He retired last year. He was actually subbing for Mr. Dickey yesterday. Okay, so he was the gentleman standing outside of Mr. Dickey's room yesterday. Okay, he's completely colorblind. All right, we actually for his retirement gift got him those glasses that allow colorblind people to see color. You should have seen when he first put them on. It was like someone slapped him in the face. He just, and unfortunately, we gave them to him in the wintertime. He was like, he couldn't wait for spring because he'd never seen green. Yeah, never seen green, had no idea. Okay, like this is like, and this is a person who doesn't know any different. Like he doesn't know that he's missing out on what green looks like. Okay, he sees everything differently than us. Incidentally, that's also why streetlights are always in the same orientation. Red's always on the top or on the left. Okay, if they're oriented horizontally, red's on the left. If they're oriented vertically, red's on the top, green's on the bottom. Always. It's for colorblind people. Okay? It's also why if you have an advanced green arrow, it doesn't just turn green, it flashes. When it turns yellow, it stops flashing. It turns solid, so that a person who's colorblind will know that it's no longer green. Because they can't tell if it's green or yellow. They can't tell the difference. Okay? So all those little things you probably didn't even think about, there's a reason for them. Okay? And a person like Mr. Van Erf, he relies on that. Okay? He looks at a stoplight and he goes, yeah, that's red. He has no idea what red is. Okay? He has no idea what green is. Okay? Can't see colors. Okay? One day he showed up and he was wearing a plaid shirt, and so was I. Mine was like red and blue, and his was like orange, or his was like green and yellow or something like that. Like they were completely opposite. He's like, hey, look! We're dressed the same today. Yeah, sure, Jerry. We're dressed exactly the same today. He had no idea. Okay? But you could also tell if he got out of the house before his wife had seen him in the morning. Because he'd be wearing two things that, like, not at all. She would have to check him before he left the house, or he'd wear, like, purple and green together. Okay? Yeah. Okay? But it is a genetic thing. Okay? And a lot of disorders are genetic. Diabetes, same thing. Okay? If you're diabetic, you have a defective form of insulin that's produced. Okay, question number 2, why is it important for organelles to have their own membranes? Compartmentalization. Okay, that's kind of the answer to that one. They can have their own environments where their process works best. Okay, what might happen if the lysome's membrane ruptured? Cell eats itself. Okay, the cell would be destroyed. Right, that's how the cell regulates or can regulate whether it is functioning properly or not. Okay, and then number 4, what problems might a cell have if all the ER was removed? Lots. Okay, I wouldn't write lots, but it would have lots of problems. It wouldn't be able to detoxify poisons. It wouldn't be able to break down complex carbohydrates. Okay, it wouldn't be able to make hormones. It wouldn't be able to repair membranes by making fats. Okay, all pretty important jobs. Basically, that's a roundabout way of asking you what does the smooth ER do or what does the rough ER do. Okay, kind of one of those roundabout ways of doing that. Okay, questions on any of that? Okay. Come up and grab a Chromebook.